one of your cells, there's a structure we talk about all the time, but maybe we take it for granted. I'm talking about the cell nucleus. But here's the thing, it's not just some boring container for your DNA. It is an absolute masterpiece of biological engineering, and it's solving what seems like an impossible problem every single second of your life. So let's jump right in and see how it works. So here's the central problem, right? Every single one of your cells has about 2 meters, that's roughly 6 feet, of DNA packed inside it. But the nucleus, the little sphere it has to fit into, is only about 6 micrometers across. That is 1,000 times smaller than a grain of sand. So the question is, how on earth does the cell pull off this incredible packaging feat?
To really get a handle on the scale of this, just imagine this for a second. Try to picture packing a rope that's 30 kilometers, or about 18 miles long, and stuffing it inside a regular soccer ball. That's the same level of compression we're talking about here. And it's not just about cramming it in. The nucleus has to keep all of that DNA perfectly organized, easy to access, and totally safe. The way it does this, well, it's a masterclass in design.
Alright, so we're going to tackle this by breaking the solution down into a few key parts. First, we'll look at how the nucleus acts like a fortress to protect and control access to all that genetic code. Then, we'll dive into its incredible multi-level filing system for compacting that code. And finally, we'll see how it actually uses all that information to basically run the entire cell. Okay, so step 1 in solving this whole packaging problem, you've got to build a secure, controlled space. And it's helpful to think of the nucleus as this kind of high-security fortress built with really strong walls and some very, very selective gates. So these fortress walls, they're called the nuclear envelope. And get this, it's not just one membrane, it's a double membrane. This creates a really clear boundary between the precious DNA on the inside and all the crazy, bustling activity of the cytoplasm on the outside. And that separation, that is absolutely the crucial first step in controlling everything.
Of course, any good fortress needs gates, right? And dotted all over this envelope, you've got thousands of these things called nuclear pore complexes. You can think of them as the cell's bouncers. They are incredibly selective. Little tiny molecules, sure, they can slip right through. But anything bigger, we're talking proteins, RNA, gets stopped at the door and has its ID checked.
So how do the bigger molecules get their VIP pass to get inside? Well, they use these special shuttle proteins called karyophorins. You've got importance, which just like the name says, grab cargo from the cytoplasm and bring it in. And then you have exportins, which do the exact opposite. They bind to stuff like RNA inside the nucleus and guide it out. It's this incredibly well-organized two-way street, and it's all tightly controlled. Alright, so the DNA is safe and sound inside the fortress. Now, for the real main event, the packing itself. The nucleus uses this mind-blowing multi-level filing system that manages to be both unbelievably dense and perfectly organized. Let's see how it's built level by level. So it all starts with your basic DNA double helix. The very first step is that this long strand gets wrapped around these little protein spools called histones. This creates a structure that looks a lot like beads on a string, and that's the nucleosome. But that's just the beginning. Next, those beads get coiled up even tighter into something we call a 30-nanometer fiber. And then, when the cell needs to divide, that fiber gets packed down even more into that classic X shape we all recognize as a chromosome. And if you want to get a look at that next level of coiling, scientists have come up with these cool models, like the solenoid and the zigzag. They're basically just theories trying to explain exactly how those beads on a string fold up into that super dense 30-nanometer fiber. It's just this beautiful, intricate piece of natural origami, and it's what achieves that incredible level of compaction.
But here's the thing, it's not just about cramming everything in there. The really crucial part is the organization. The nucleus is smart about it. It arranges DNA based on how often it needs to be used. The stuff that's less compact and easy to get to, that's called euchromatin. Think of it as the active, open-shelf section of a library. And the really dense, tightly packed DNA, that's heterochromatin. That's like the deep storage archive, holding all the genes the cell doesn't need very often.
Okay, so we've got the DNA protected. We've got it organized. Now what? Well, now the nucleus can get to its real job, acting as the cell's command center. It reads the genetic blueprints to build, run, and maintain the entire cell. And right smack in the middle of this command center, you've got a structure called the nucleolus. And it has one absolutely essential job. It is the cell's dedicated ribosome factory. And why is that such a big deal? Well, ribosomes are the little machines that actually build all the proteins in the cell. So think about it. If you don't have the nucleolus making ribosomes, you can't make any proteins. No enzymes, no hormones, no structural parts, nothing. The entire production line of the cell just grinds to a halt. The nucleolus literally builds the builders.
You know, when you boil it all down, maybe the nucleus's greatest trick is just this: separation. By keeping transcription, that's the process of reading DNA and RNA, safely inside its walls, while letting translation, making proteins from that RNA, happen outside in the cytoplasm, it creates all these checkpoints. This allows for really complex gene regulation, letting the cell fine-tune the genetic message before it becomes a final product. It's brilliant. So we've seen how elegant and incredible this whole system is. But what happens when it goes wrong? When this perfect little machine breaks, the consequences can be, well, they can be devastating. So what holds the nucleus together, gives it its shape? It's this protein mesh called the nuclear lamina. Now, if the genes that code for these lamina proteins have a mutation, it can cause a whole group of diseases called laminopathies. And what happens is the nucleus itself becomes misshapen and just doesn't work right.
And a really tragic but powerful example of this is a condition called progeria. It's a rare genetic disorder caused by a tiny defect in a single one of those lamin proteins. This leads to unstable nuclei, and it causes children to age incredibly rapidly. It's just a devastating reminder that the physical structure of the nucleus is directly tied to our health, and even the process of aging itself. And it's not just the lamina that can cause problems. In autoimmune diseases like lupus, the body's own immune system starts attacking its nuclei. If cell division goes wrong, you can end up with cells that have no nucleus at all, or even multiple nuclei. And then you've got viruses like herpes virus, which have figured out how to invade the nucleus and hijack its machinery for their own replication. So when you think about it, the nucleus is this fortress, this library, this command center, all rolled into one, controlling the life and destiny of the cell. But what happens when the cell gets an order for its own destruction? When it's told to deliberately and systematically tear down this incredible structure from the inside out? Well, that's a whole other story.